There is a particular kind of quiet that exists late at night. Not the absence of sound exactly, but a thinning of it. The world outside does whatever it does without you for a while. And in that thinning, in that small recession of the day's noise, something becomes possible that wasn't quite possible before. You can follow something small. You can stay with something that would normally slip past you unnoticed. Welcome to the Sleepy Biologist. If you've been here before, it's good to have you back in this space. If tonight is your first time, then you've arrived somewhere that moves slowly on purpose. This channel exists because biology, real biology, the kind happening inside living things at every hour, is one of the quietest and most extraordinary things there is. And the members who support this channel make it possible to keep going deeper, keep finding the threads worth following into the dark. If you've ever hit super thanks, or joined as a member, or simply left a comment about where you were listening from, thank you. Genuinely, those things matter more than they probably seem. Tonight we're going somewhere that most people will never think to go. We're going underground, not deep underground, not far, just beneath the surface of a field or a forest floor, or the dry soil beside a road you've probably walked past without a second thought. Down into a network of chambers and tunnels that has been there, in various forms, for longer than almost anything else on Earth. Down into a colony. And the first thing you notice, if noticing is even the right word for something this subtle, is that the air is different. You've probably felt this before, the way certain spaces have their own atmosphere before you understand why. A room that's been closed too long, a kitchen where something was cooked hours ago, a place where many bodies have been. The sensation arrives before any explanation does, and your own body has already begun responding before your mind catches up. Inside an ant colony, the air is doing something like that, constantly. Every ant in the colony is producing chemical compounds from glands distributed across its body. Some at the head, some along the thorax, some near the abdomen. These compounds evaporate into the tunnel air at different rates. Some disperse quickly, lasting only seconds. Others linger, coating the walls of the nest, settling into the substrate, becoming part of the ambient chemistry of the place in a way that's almost structural. The colony smells the way it does because of what every individual member has been contributing moment by moment, without pause. And that smell is not decoration. It is not incidental. It is information. When you walk into a familiar place and feel, before you've consciously registered anything, that something is slightly off, that is your nervous system responding to sensory input before language catches up. Ants live in a version of that experience that never resolves into language at all. They exist permanently inside a world of chemical signal, where the information is continuous and ambient and everything. The compounds drifting through nest air carry data about food sources, about danger, about reproductive status, about where to go and how fast to get there. They carry something else too, something quieter, something that runs underneath the more urgent signals the way a low frequency runs beneath sound you can consciously hear. They carry information about health. An ant that has been exposed to a pathogen, a fungal spore, a harmful bacterium, a microorganism that has landed on its cuticle, begins to emit a slightly altered chemical signature, not dramatically, not in a way that announces itself. The change is subtle, a shift in ratio rather than a new compound entirely, the chemical equivalent of a familiar voice speaking at a slightly different register. But the colony detects it. This is worth staying with for a moment, because it happens before any visible symptom appears, before the exposed ant moves differently, before it grooms itself differently, before anything you could observe with your eyes has changed. The air inside the colony is already updated. Other ants have already begun processing that altered signal. their antennae reading the change, their behavior beginning to adjust in ways so slight they would be nearly impossible to document from outside. The infection management system of an ant colony does not begin with a response. It begins before that. It begins in the air, in the ambient chemistry of a space where information and atmosphere are the same thing. There are parts of your own body that operate this way, not through thought, not through conscious detection, but through systems that have been reading your internal state continuously since long before you were born. Your immune system is not waiting for you to notice something is wrong. Processes of surveillance and identification and early response are already running at a level you will never directly feel. It is possible to find something strangely settling in that fact if you let it sit for a moment. The colony does not need to understand what it is doing to do it extraordinarily well. Somewhere in a tunnel right now, below ground that is probably cold, in air that carries more information than most sensory systems could ever process, an ant is returning from the surface. It has travelled a distance that, relative to its body size, is remarkable. It is carrying something or it isn't. It is chemically intact or it isn't quite. The colony is already beginning to know. The antennae of the ants it passes graze its body in brief contact, a touch that lasts a fraction of a second, but transfers chemical data across surfaces with a fidelity that is genuinely difficult to fully account for. That information moves inward, from ant to ant, deeper into the nest, spreading through the population not like a message being passed, but like a change in the air itself. This is not the most dramatic part of what ants do. What makes it worth staying with is how ordinary it is for them, how constant, how unhurried, how entirely woven into the structure of every moment inside the colony. The air is always carrying something. Something is always being detected. And somewhere between detection and response in that small biological interval, a decision of sorts has already been made. Not by any individual ant. Not by the queen who is elsewhere, producing eggs and the particular pheromones that signal her presence to the colony. By the system, by the chemistry of the space itself. The nest air settles around all of this the way temperature settles into a room, and not pushed, not arranged, simply present. And the colony continues, breath by chemical breath, doing what it has been doing for tens of millions of years. Tonight, that is enough to begin with. There is more to find down here. Deeper in the tunnel, in the chambers where the colony does its quietest and most careful work, something has been running for longer than most living systems have existed. We haven't reached it yet, but the air inside is already carrying the first trace of what it is. Think about what it means to be very small. Not small in the way we sometimes imagine smallness. Fragile, quiet, easily overlooked. Small in the way that changes the rules entirely. Small in the way that makes certain problems enormous and certain solutions unavailable. An ant weighs somewhere between 1 and 5 milligrams depending on the species. Its entire body fits inside a space you could cover with your thumbnail. And inside that space it runs, it lifts, it navigates, it detects chemical signals in concentrations so low that measuring them requires instruments that didn't exist a century ago. It does all of this without stopping, and it cannot get sick, not the way you can. You have options when your body encounters a pathogen, not comfortable options, but options. You have a fever response that raises your internal temperature to a point where many microorganisms struggle to replicate. You have a lymphatic system moving immune cells through fluid channels that run parallel to your bloodstream. You have organs dedicated to filtering, to memory, to escalating response when the first response isn't enough. An ant has none of that, not in the same sense. What it has instead is something worth sitting with for a moment, because it's genuinely strange when you look at it directly. An ant's immune system is simple compared to yours. Dramatically so. It has hemocytes, cells circulating in its hemolymph, the fluid that fills its body cavity and does the work that blood does in you. And these cells can engulf foreign particles, can respond to certain signals, can mount a basic internal defense. But the system is not layered. It does not escalate through stages the way yours does. It does not carry immunological memory in the same way. Once something gets through the cuticle, through the hard external surface that is the ant's first and most important barrier, the options narrow quickly. This is why the outside matters so much. The cuticle is not just structural, it is the primary defense. A waxy, chemically complex layer that resists moisture loss, provides physical protection, and carries compounds that have antimicrobial properties in their own right. The chemistry of the cuticle varies by species, by caste, by age. It is not static. It changes in response to what the ant encounters, absorbs secretions from nestmates during grooming, accumulates compounds from the nest environment, but it can be breached. Fungal spores are particularly good at this. Certain species have evolved specifically to exploit the moment of entry, producing enzymes that degrade the cuticle's surface, creating a pathway inward. Once inside, the consequences move quickly. The ant's internal defenses activate, but the window is narrow. It's not entirely clear how narrow. That uncertainty is real. Researchers are still working out the precise timelines for different pathogens in different species under different conditions. What is clear is that the colony cannot wait for individual ants to mount internal defenses alone. The biology suggests the whole system was never designed to work that way. The individual is not the unit of defense, the colony is, and so the body of the individual ant exists in a kind of permanent biological tension. It is doing enormous things relative to its size, genuinely enormous things, on a continuous basis. Foragers travel the equivalent of several kilometers in human-scale terms during a single day. Their muscles work hard, their nervous systems process constant sensory input. Their cuticle is in contact with soil, with plant surfaces, with other insects, with the open air, which carries its own invisible load of microorganisms at all times. All of that exposure, all of that physical cost. And if something gets in, the options are limited. You've probably felt the early edge of your own immune system activating. That specific tiredness that isn't quite fatigue, the slight ache that settles into joints before a fever develops, the sense that your body has quietly redirected resources towards something internal without consulting you. There is something almost uncomfortable about how recognizable that feeling is once you know what it means, your body pulling inward, priorities shifting. An ant cannot pull inward. There is no mechanism for rest in the same sense. The colony's needs continue. The ant continues. What changes instead is the chemistry happening at its surface and in the air immediately around it. The colony begins to respond before the ant can. That is the architecture this chapter is pointing toward, not the individual body's defenses, though those exist and matter, but the way the colony itself functions as a kind of distributed immune organ. Each individual ant is vulnerable in ways that a single body cannot fully compensate for. What compensates instead is the system that surrounds it. This is not a metaphor. It is a structural biological reality. The colony distributes immune function across the population the way your body distributes it across organs and cell types. No single component handles everything. No single component could. The smallness of the ant is not just a physical fact, it is the condition that made this entire system necessary. And it has been necessary for a very long time. How long exactly is one of those numbers that loses meaning if you say it quickly? Tens of millions of years is the honest answer. long enough that the biology being described here is not an adaptation to recent conditions. It is a response to pressures that predate the existence of most things currently living on Earth. The ant running through the tunnel right now, its antennae sweeping, its cuticle reading the chemical atmosphere of the nest, its hemolymph carrying its simple complement of immune cells. It is a product of all of that time. It cannot afford to get sick. So far, for most of them, most of the time, they don't. What keeps that balance is not inside any one ant. It lives in the space between them, in the touching of antennae, in the grooming, in the compounds that move from body to body through contact so brief it barely registers as contact at all. The individual is small, the system built around its smallness is something else entirely. We haven't reached the centre of it yet. There is a number that appears in the research literature on anti-evolution that tends to stop people for a moment when they encounter it properly, not as a figure to memorize, but as a duration to actually consider. 130 million years. That is the approximate age of the earliest ant lineages, the point at which something recognizably ant-like was moving through a world so different from the current one that almost nothing living today would have been there to see it. The flowering plants were new, the dinosaurs were still present. The continents were arranged differently. The climate was different. The atmosphere carried a different ratio of gases through air that no human lung has ever breathed. And in that world, in soil that no longer exists, in tunnels pressed into ground that has long since shifted and compressed and become something else entirely, ants were already encountering fungi. Not the same fungi that exist today. Evolution does not hold still, but the relationship, the ancient, persistent, biologically consequential relationship between ant bodies and fungal spores was already underway, already exerting pressure, already beginning through the long and impersonal mechanism of differential survival to shape the chemistry of the ant cuticle, the behavior of the colony, the glands that would eventually produce compounds that function, in effect, as antibiotics. I tend to think that this is one of the most quietly remarkable facts in all of biology, though I recognize that is a personal response to something that simply is what it is. The biology does not require anyone to find it remarkable. It continued without observation for the vast majority of its history, operating in the dark underground in colonies that lived and died and left almost nothing behind except the slow accumulation of change in the genomes of their descendants. What we have now is the result of all of that. The metapleural gland, a structure found on the thorax of most ant species, absent in some, modified in others, produces a secretion that has been the subject of serious scientific attention for several decades now. The secretion contains compounds including phenylacetic acid and various other organic acids that suppress the growth of bacteria and fungi on the ant's body surface and in the nest environment. It is applied during grooming, distributed through contact, spread across the walls of the nest in the normal course of colony movement. It functions the way an antibiotic functions, not through conscious application, not through any understanding of what it is doing, but through chemistry that works because the lineages that produced it survived better than the lineages that didn't. Alexander Fleming observed in 1928 that a mold, Penicillium, was producing something that killed the bacteria around it. He recognized the significance of that observation, and what followed from it changed medicine in ways that are difficult to overstate. But the principle that one organism could produce a compound that suppressed another was not new when Fleming encountered it. It was not new by a measure of time that makes the entire history of human civilization feel like a recent afternoon. Ants had been running that principle continuously in the dark for somewhere between 30 and 100 million years before a human scientist looked at a contaminated petri dish and understood what he was seeing. This is where the extended metaphor this chapter has been quietly building needs to be gently set down, because the comparison between Fleming's penicillin and the ant's metapleural secretion is useful only up to a point, and that point has now been reached. Fleming's discovery was an observation of something a fungus was doing for its own competitive reasons. The ant's antibiotic system is something the ant produces itself in a gland that evolved specifically for that purpose, distributed through behavior that reinforces its effect. The parallel is instructive. It is not precise, and precision matters more than a clean comparison when the biology is this interesting on its own terms. So set the comparison aside. What remains is this: a gland, small enough that it requires magnification to examine, sitting at the posterior corner of an ant's thorax, producing a secretion that the colony depends on in ways that took researchers a surprisingly long time to fully appreciate. Early studies established the antimicrobial properties. Later work began to reveal how the distribution worked, how grooming behavior spread the compounds, how the nest walls accumulated a chemical environment that was actively hostile to certain pathogens, how the behavior of the colony and the chemistry of the individual were not separate systems but one continuous biological process operating at two scales simultaneously. The past is present in all of this in a way that is worth acknowledging before moving on. The gland exists because of pressure applied by organisms that no longer exist in their original form, in environments that have been geologically transformed, across timescales that human cognition is genuinely not well designed to inhabit. And yet the gland is there, in the ant moving through the tunnel right now, producing what it has always produced, maintaining what it has always maintained. There is something in evolutionary biology that functions like memory without being memory. The accumulated record of what worked, written not in neurons but in the sequence of functional genes, expressed in proteins, realized in structures like this small gland doing its quiet chemical work. It is not memory. I find it difficult not to reach for that word anyway, and then pull back from it, because the accuracy matters. It is older than memory. It is the thing that made memory eventually possible. The colony underground is not thinking about any of this. It is doing what it does. Moving, building, grooming, foraging, tending, and in the doing of it, the antibiotic system runs. It ran this morning. It ran in the middle of the previous night. It ran while you were doing something else entirely, while you were going about the ordinary business of your day, in a world built on the surface of a planet that has been hosting this chemistry for longer than it has hosted almost anything else currently alive. The air in the tunnel carries the faint trace of those compounds now. The walls hold them. The bodies of every ant in the colony are coated in them, transferring them to every surface they touch, to every nestmate they groom, to the eggs and larvae in the chambers below. It began before the word fungus had any meaning to anyone. It is still beginning in a sense. That is where the chapter started, and it is quietly where it ends. Watch two ants meet, not with urgency, just watch. They approach each other along a tunnel wall, antennae moving, bodies angling slightly, and then, briefly, almost too briefly to register, one runs its mouth parts along the body of the other. A pass across the head, along the thorax, sometimes the abdomen, and then they separate, each continuing in whatever direction it was already going, as though nothing particular happened. It is easy to read that as social, as care, as something warm between small creatures sharing a life underground. And you start to notice when you sit with it long enough that the reading isn't entirely wrong, but it is incomplete in ways that matter. What just happened was chemical. An ant's mouthparts are not simply mechanical tools for moving food, they are lined with glands. The saliva produced during grooming contains compounds, enzymes, antimicrobial secretions, components of the metapleural gland transferred through body contact that are applied to the surface being groomed with a consistency and coverage that is, when you look at the data, genuinely precise. The ant being groomed receives a chemical treatment across its cuticle. Fungal spores present on the surface are exposed to compounds that interrupt their germination. Bacterial loads are reduced. The cuticle itself is maintained in a condition that resists future contamination more effectively than an ungroomed surface would. This happens dozens of times per day for each ant in the colony. It happens faster when a nestmate has been outside. Faster still when something in the returning ant's chemical signature has shifted. When the ambient signal carried on its body suggests exposure to something the colony's chemistry recognizes at some functional level as a threat. The grooming accelerates before any visible sign of infection appears. Sit with that for a moment. Not the efficiency of it, though the efficiency is real, something quieter than that. The fact that the response begins in a window of time before the problem has fully declared itself, before the fungal spore has breached the cuticle, before the internal cascade of infection has started, before anything has gone wrong in any way that could be observed from outside. The colony is responding to a probability, to a shift in chemical ratio that says something may be different here. The grooming is an intervention in a future that hasn't happened yet. This is where the soft cluster of comparison dissolves because no comparison fully holds here. There is no human equivalent that maps cleanly onto what is being described. Your own immune system runs surveillance continuously, but not through the action of other people touching you. Your skin provides barrier protection, but it is not chemically refreshed by social contact. The biology of the ant colony is doing something that doesn't have a clean analogue in your body or your experience, and it's worth letting it be its own thing, rather than reaching for a translation. So just observe. In the deeper chambers of the nest, where the larvae are kept, grooming is near constant. The nurse ants, workers whose behavioural role keeps them inside the nest rather than foraging, move between larvae with a regularity that, in time-lapse footage, looks almost rhythmic. They are applying antifungal compounds to surfaces that cannot protect themselves. The larvae have no cuticle in the adult sense. They are soft, permeable, vulnerable to the same fungal spores that drift through the nest air and settle on every surface. Without continuous grooming, the larval mortality rate would be different. The research on this is clear. The colony grooms its young the way your body maintains barrier function in tissues that cannot maintain themselves. Not through choice, not through understanding, but because the colonies that did it less carefully are not here to study. This is a quiet fact. It doesn't announce itself. It feels like it should be larger than it is somehow. The discovery that grooming in ant colonies functions as a distributed antimicrobial system, that the mouth of one ant is delivering chemical protection to the body of another in a way that measurably reduces infection rates, is the kind of finding that, when you encounter it plainly, produces a small internal stillness. It's easy to forget that this is happening right now, in the ground beneath ordinary places, in the dark, continuously. The ant that groomed another ant a moment ago is already moving on. It does not know what it did. There is no knowing involved in any of this. Not in any sense of the word that applies to awareness or intention. The behavior exists because it worked, because it reduced losses in colonies that expressed it more fully, because over the span of time described in the previous chapter, that span so long it loses texture, the ants that groomed carefully left more descendants than the ants that didn't. And so here it is, in the tunnel, in the brief pass of mouth parts across a thorax, in the transfer of compounds from one body to another, so fast the exchanger was over before you finished registering it. You've probably felt the specific sensation of being cared for without the other person making anything of it. Someone adjusting something near you without comment. A small act that requires no acknowledgement because it wasn't offered as something to acknowledge. The ant being groomed experiences nothing like that. There is no experience of care involved, no social meaning being exchanged. But the biology produces something structurally similar. Protection delivered through contact, quietly in passing. The tunnel is still. Somewhere in it, antennae are moving. Somewhere, two bodies are briefly in contact. Something is being transferred. The compound lands on the cuticle. The spore, if there is a spore, encounters a chemical environment it cannot easily survive. The ants separate. Both continue. In the chamber below, a larva is being tended. The nurse ant's mouthpart to move in the same brief practiced way. The same compounds are applied to a different surface in a different concentration in a slightly different formulation that the nurse ant's glands produce in response to the specific chemical signals of the larval environment. The adjustment is automatic, the precision is real. There are glands involved in this that researchers are still characterizing. Not everything is mapped. Not every compound has been fully identified. The system is understood well enough to describe, not well enough to fully explain, and that gap, that honest, productive gap between description and explanation, is where a great deal of current research on ant biology quietly lives. It's easy to forget that not everything has been found yet. Some of what the grooming is doing is still being discovered in laboratories where researchers are separating chemical fractions and testing them against fungal cultures and slowly building a picture of something that has been working, without any assistance from science, for tens of millions of years. The picture is incomplete. The grooming continues anyway. Below the grooming behavior, below the chemistry of the saliva, below the compounds being applied to cuticle surfaces across the colony right now, there is a gland. Small, paired. Sitting at the base of the thorax in most ant species. It has been mentioned before briefly. It deserves more than that. The next chapter stays there. It sits at the posterior lateral corner of the thorax, one on each side, small enough that early anatomists working without adequate magnification missed it entirely or noted it without understanding what they were looking at. A paired structure so modest in its physical dimensions that its significance took a surprisingly long time to become clear, even after it was properly identified and described. The metapleural gland. In the taxonomy of ant anatomy, it occupies a position that is both unremarkable, just another structure among the many structures packed into a body plan refined across geological time, and quietly central to almost everything this video is moving toward, because without it, or rather without what it produces, the colony's relationship to infection would be fundamentally different, and the story of how ants arrived at something functionally equivalent to pharmaceutical intervention would have no mechanism to point to. It is worth staying with the gland itself for a moment before moving into what it does, the way you might look at an ordinary object for longer than usual and find that it becomes less ordinary the longer you look. The gland is exocrine. It secretes outward, onto the body surface rather than into the ant's internal system, which is itself a structural choice worth noticing, because it means the gland's output is designed to operate at the boundary between the ant and the world, at the surface where contamination arrives and where the first line of defense therefore needs to be. The secretion accumulates in a reservoir associated with the gland and is released, either continuously at low levels or in higher concentrations during grooming, and in response to specific stimuli, across the ant's cuticle and into the immediate environment of the nest. What the secretion contains varies by species, and this variation is one of the more interesting threads in the current research. because it suggests that different lineages of ants have arrived at different chemical solutions to the same fundamental problem, tuning the output of the same basic glandular structure to the specific microbial pressures of their particular ecological niche. In many species, phenylacetic acid is a significant component, a compound with documented antifungal and antibacterial properties that, when applied to a surface, creates conditions hostile to the germination of fungal spores and the replication of certain bacteria. There are indole compounds. There are fatty acids. There are components that researchers are still in the process of identifying and characterizing, which is not a caveat meant to undermine confidence in what is already known, but rather an honest description of where the science currently sits, in productive incompleteness, with more structure visible than a decade ago and more structure still waiting to be resolved. The part that stands out most when you read through the literature on this gland is not any single compound, but the relationship between the gland's output and the ant's behavior. The two are not separate. Grooming distributes the secretion. The structure of the nest, the way ants move along particular paths, the way they groom larvae in specific locations, the way workers returning from outside are met and attended to near the nest entrance rather than deep inside. All of this behavioral architecture serves, among other things, to distribute the metapleural secretion in a pattern that provides the most coverage where contamination risk is highest, like a system of resource allocation that has no planner and no plan, but produces a consistent and functional output anyway. Still, there is something the gland does that is not fully accounted for by the identified compounds and the documented behaviors. A residual effect visible in experimental data that doesn't resolve cleanly into the current mechanistic picture. And the researchers who work in this area tend to note it with the particular care that scientists use when they are pointing at something real that they cannot yet fully explain. Some ant species have lost the metapleural gland across evolutionary time, and the pattern of those losses is itself informative, because the losses cluster in lineages that have moved away from soil nesting into drier or more arboreal environments where fungal pressure is different, or into social structures where the colony's other antimicrobial behaviors have become sufficiently robust to partially compensate for the gland's absence, which suggests that the gland is not irreplaceable in absolute terms but is deeply load-bearing in the ecological context where it is retained. The army ants have reduced or lost it. The carpenter ants have it. The leafcutter ants have a version of it that appears to have been modified in ways consistent with the extraordinary fungal management demands of their agricultural lifestyle, which is itself a story worth following, though this chapter is not quite the place for that yet. What this chapter is a resting point, a place to stay with the fact that a structure this small, paired glands at the base of a thorax, in a creature weighing a few milligrams, is doing something that the colony depends on in a fundamental way, that the science of what it does is real and documented and still unfinished, and that the unfinished quality is not a weakness in the story, but an accurate reflection of where living biology actually is when you look at it closely enough. Your own body contains structures whose full function remains, after decades of careful research, not entirely resolved, glands and tissues and cellular populations that do known things and possibly other things, organs whose secondary roles are still being characterized, interactions between systems that produce effects that are real in the data and not yet fully mechanistically explained. The metapleural gland is in good company in that sense, residing in the long middle ground between discovery and complete understanding where most of actual science spends most of its time. The gland is working right now, in tunnels below ground, in ants that are moving or grooming or standing still in the particular way ants sometimes stand still, that looks like nothing and maybe nothing or maybe something that hasn't been studied yet with sufficient resolution to know. The secretion is coating cuticle surfaces, It is settling into the substrate of the nest walls. It is being transferred in trace amounts through every grooming contact and every brief antennal touch between nestmates, diffusing through the colony's social network the way something important diffuses through any system where everything is in contact with everything else. It began working long before the first researcher identified the gland. It will continue working regardless of how much of its mechanism is eventually understood. That is, in the end, what stands out most about this small structure. Not its elegance, not its complexity, but its reliability across a span of time that makes the entire history of human science look like the very beginning of an attempt to notice something that was already, quietly, thoroughly underway. The gland secretes. The colony distributes. The infection, in most cases, does not take hold. What happens when something does get through? When a spore lands at the wrong moment in the wrong concentration and the chemistry at the surface is not quite enough, is where the next chapter begins to look. A single spore, that is where this starts. Not with a swarm of them, not with a catastrophic exposure, but with one, microscopic, drifting, carried on air currents that move through the tunnel entrance the way air moves through any opening. Not dramatically, just continuously, because air moves through openings and carries what it carries. The spore lands on the cuticle of a forager returning to the nest. The forager doesn't stop, doesn't register anything different. Why would it? The landing of a spore produces no signal the ant's nervous system is equipped to detect directly. There is no sensation involved, no pain, no pressure, nothing that functions like an alert. The spore is simply there now, on the surface, held by the slight adhesive quality of the cuticle's waxy outer layer, waiting in the only way something without intention can wait, which is by existing. By being present, by beginning, if conditions allow, the process it was built to begin. You know the feeling of something happening in your body before you have any conscious awareness of it. Not always, but sometimes. The slight heaviness that precedes a fever, the faint shift in how your throat feels hours before it becomes sore, something moving in the system before the system announces itself. It is a strange feeling when you notice it, the sense of being slightly behind something that is already underway inside you. The ant has no version of that feeling, but the colony does, not as sensation, as chemistry, as the altered signal profile of a returning forager that has been in contact with something the nest air doesn't usually carry in that concentration. The ants near the nest entrance are the first to read it. Antennae touching the forager in passing, mouthparts briefly grazing its surface during the rapid, almost reflexive grooming that happens at the threshold between outside and inside. This boundary grooming is not casual. It is, in terms of what it accomplishes biologically, one of the most consequential behaviors in the colony's infection management system. Ants returning from the surface pass through a zone of intense chemical attention before they move deeper into the nest. The workers stationed near the entrance are not there by accident. Task allocation in ant colonies is not random, and the ants near the entrance tend to be those whose chemical profile and behavioral repertoire make them suited to that specific role. They are, in a functional sense, the colony's first detection layer. The grooming they perform on returning foragers applies metapleural secretion to the cuticle surface. It removes particulate matter. It exposes any spores present to compounds that interrupt the germination process, the biochemical sequence a fungal spore must complete in order to penetrate the cuticle and begin growing inside the ant's body. Whether a spore germinates depends on timing. The window between landing and penetration is narrow for most fungal pathogens that target ants. The spore must adhere, produce the right enzymes, degrade the cuticle at a rate fast enough to establish entry before the ant's surface chemistry disrupts the process. The metapleural secretion doesn't need to kill the spore outright. It needs only to slow the germination process enough that the window closes before penetration occurs. Often. That is what happens often, not always. There is something clarifying about that honest qualification, actually. The system is not perfect. It was never selected for perfection. Evolution doesn't work toward perfection. It works toward good enough under specific pressures, and good enough here means most spores, most of the time in most conditions, do not successfully penetrate. The fraction that do is small. The colony absorbs that fraction through other mechanisms, but the fraction exists, and pretending otherwise would be a cleaner story than the biology actually tells. The forager moves deeper into the nest now. It has been groomed at the entrance. The spore load on its surface has been reduced. Whether the specific spore that landed, the one this chapter began with, has been reached by those mouth parts, whether the compound concentration at that exact location on that exact patch of cuticle was sufficient, is a matter of geometry and timing and the particular chemistry of this individual ant on this particular day. Biology operates at that level of specificity constantly. It is easy to describe systems. It is harder to hold in mind that every system plays out inside individual bodies, one body at a time, where the outcome is particular rather than statistical. The forager settles into the nest. The colony around it continues. Workers move past, some make antennal contact, some don't. The chemical signal the forager carries, slightly altered, subtly different from a nestmate that has been inside all day, continues to be read by each ant it encounters, and each reading is a small input into the distributed calculation the colony is running about whether this individual requires more attention. More grooming may come from nestmates deeper in the colony who detect the same signal. The intensity of grooming an ant receives correlates in the research with the degree to which its chemical profile has shifted away from the colony's baseline. This is not a decision any ant makes. It emerges from the aggregate behavior of a population each member of which is responding to local chemical information. The spore, if it is still viable, is waiting. Your body does something similar with threats that have crossed an initial barrier, not ignoring them, not yet mounting a full response, but running a quiet background process of detection and low-level engagement while the situation resolves itself one way or another. There is a biological patience to early immune response that is often invisible from the inside until it tips into something noticeable. The ant's cuticle is that first barrier. The entrance grooming is the secondary pass. What comes after, if something gets through both, is different in character, less ambient, more targeted, and involving behaviours that the colony reserves for situations that have moved past the threshold of chemical management. But that threshold has not been crossed yet. The nest is calm. The air carries its usual chemistry. The forager is still. Whatever is on its surface is either losing viability or not. And the colony is still reading, still touching, still applying what it has always applied to surfaces that need it. Somewhere in this, in the quiet interval between exposure and outcome, is where most of the biology actually lives. Not in the dramatic cascade, not in the failure, in the long, unremarkable middle where the system runs and the spore sits, and the colony continues without knowing that it is, right now, in the process of handling something. It handles things like this continuously. Most of them never become anything at all. The forager rests. Another ant passes close, antennae briefly extended. Contact, grooming, the familiar compounds moving from one surface to another in the dark. Outside, above the tunnel entrance, the world continues. Spores drift on an air that moves without direction. Other foragers are returning. The threshold between outside and inside is being crossed again and again, by bodies that have been in contact with everything the surface world carries. The colony receives them all, and somewhere in a deeper chamber, in a part of the nest the forager hasn't reached yet, a different process runs, slower, older, more chemically elaborate, that exists precisely for the cases where what just happened at the gate was not quite enough. There is a behaviour in ant colonies that researchers initially struggled to categorise, not because it was rare, because it was common enough to be documented across dozens of species, consistent enough to suggest deep evolutionary origin, and uncomfortable enough in its implications that the language used to describe it has shifted several times as the understanding has deepened. An ant leaves the nest, not to forage, not along a trail, away from the colony, away from nestmates, into open ground that offers nothing a healthy ant would seek out. It moves with a directedness that looks purposeful and is, in some functional sense, purposeful, but the purpose belongs to the colony, not to the ant. It does not return. Your body does something structurally similar, though you never feel it directly. Cells that have become dangerous, infected cells, cells whose internal machinery has been hijacked by a virus, cells that have begun to replicate in ways that threaten surrounding tissue, undergo a process called apoptosis. Programmed death. The cell dismantles itself from the inside, packaging its contents carefully so that the breakdown products don't damage neighbours, signalling its own end through biochemical sequences that have been conserved across enormous spans of evolutionary time. The cell does not resist. The ant does not resist either. What triggers the departure is chemical. An ant whose infection load has crossed a threshold, whose internal hemolymph chemistry has shifted, whose surface compounds have changed in ways that nestmates continuously read, begins to receive signals from the colony that alter its behaviour, or it generates those signals itself from internal changes that modify its own behavioural outputs without any external instruction. The research on this is still being refined. What is clear is that the departure is not random. It is not disorientation. Ants that are experimentally infected with certain fungal pathogens show a statistically significant tendency to leave the nest and remain outside even when they retain the physical capacity to return. They are not lost. They are, in some sense that doesn't require awareness to be real, removing themselves from the population. Sit with the geometry of that for a moment. The colony's infection management system operates on a principle that individual medicine cannot use. It can afford to lose components in order to protect the whole, not wastefully, not casually. The threshold for self-removal or enforced removal is set by the biology at a point that balances individual loss against collective risk, and that balance has been calibrated across millions of generations of colonies that set it differently and paid different prices. The colonies that exist now set it about right. What about the ants that are removed by others rather than leaving voluntarily? This happens too. Nestmates detect the chemical signature of advanced infection and engage in behaviours ranging from intensified grooming, a last attempt at surface decontamination, to active removal of the infected individual from the colony interior. The line between these two responses, and where the biology tips from one to the other, is not fully mapped, and the tipping point likely varies by species, by pathogen, by the current infection pressure the colony is experiencing. There is no moment of decision. No individual ant weighs anything. The behaviour emerges from chemical gradients, from the aggregate responses of a population each member of which is doing something local and simple. What looks from outside like a collective judgment is the output of many small biological events aligning in the same direction. Humans tend to impose narrative on this. The sacrificial ant, the hero of the colony, the individual giving itself for the greater good. These framings are not biologically wrong in every respect. The outcome does benefit the colony at cost to the individual, but they import a set of assumptions about agency and meaning that the biology does not support. The ant is not giving anything. It is behaving in accordance with its current chemical state, which has been shaped by the infection and by the signals it is receiving from its environment, and the behavior that results happens to serve the colony's interests. That is a less comfortable framing than sacrifice. It is more accurate. There is something in your own body's systems that operates with a similar indifference to the individual component. The immune response, when it escalates, does not negotiate with infected cells. Apoptosis does not offer the cell an alternative. The biochemical signals that initiate programmed death are not a judgment. They are a process, running along pathways laid down long before any individual body existed to run them through. The ant outside the nest is sitting in open air now. It may groom itself. The behavior persists even in isolation, even when there are no nestmates to receive the chemical information that grooming produces. Some behaviors run because they run, because the neural circuitry that generates them is still active, and the stimulus conditions that suppress them are absent. The colony inside continues. The queen produces eggs, workers tend larvae, foragers return through the entrance, are groomed at the threshold, move deeper into the nest carrying food and chemical information. The system has already adjusted for the absence of one component, not consciously, through the redistribution of task allocation that happens continuously as the colony's population composition shifts. One fewer forager, the trail chemistry adjusts. Other foragers cover the gap. The nest does not register the loss the way a human group registers the loss of a member. There is no equivalent of grief in the colony's chemical vocabulary, no signal that means absence in the way absence means something to beings for whom individual relationships carry weight. The ant outside moves less now. The infection is progressing. The fungal hyphae, if this is a fungal infection, are extending through the soft internal tissues, consuming resources, producing compounds that alter the ant's behavior in ways that may serve the fungus's own reproductive interests. A different story and a darker one that runs alongside this chapter, without quite entering it. What enters this chapter is simpler. A body. Outside. The colony intact. Inside. The distance between them. A few centimeters of soil and tunnel wall functioning as the margin the colony required. The margin held. In the chamber where the larvae are kept, a nurse ant moves between rows of pale, soft bodies, mouthparts working, applying the same compounds that have always been applied, maintaining the surface chemistry that keeps the most vulnerable members of the colony below the threshold where infection can take hold. The work is the same as it was this morning. It will be the same tonight. The ant outside the nest is still now. The biology is doing what biology does at the end of a body's function, not ending so much as converting, matter moving into different forms, the same atoms that were an ant, becoming part of the soil that the colony has always lived inside. A forager will pass near this spot tomorrow. It will not pause. Below ground, in the dark, in the chamber where the queen rests surrounded by the workers who tend her continuously, something is being maintained that the biology has always prioritized above everything else, the colony's continuity, not any individual within it. What that continuity depends on, the chemistry, the behavior, the glands, the grooming, the quiet removal of what threatens, runs without pause in the tunnels below, and the next chamber deeper holds something that complicates the picture in a way this chapter has only begun to gesture toward. Here is something that takes a moment to fully land when you first encounter it. Leafcutter ants are farmers, not metaphorically, not approximately. They cultivate a specific fungus, tend it across generations, protect it from competing organisms, and depend on it so completely that neither the ant nor the fungus can survive without the other. This relationship is somewhere between 50 and 60 million years old. The fungus the ants grow today is not the same fungus their ancestors started with. It has co-evolved with the colony over that entire span, shaped by the same pressures that shape the ant, until the two organisms have become something neither would be without the other. That alone is worth a moment. But the part this chapter is interested in sites inside the garden itself, in the problem that any farmer, any organism that cultivates another living thing in a contained space, inevitably faces. Contamination. When you grow something in an enclosed environment, you create conditions that favour growth. And growth conditions do not discriminate. The warmth, the humidity, the nutrient density of a leafcutter fungus garden are not welcoming only to the cultivated fungus. They are welcoming to everything that competes with it. Foreign mold spores that drift in on returning foragers, bacteria that thrive in the organic richness of chewed leaf substrate, microbial hitchhikers that arrive continuously from the surface world the ants move through every day. A leafcutter garden without active protection would not stay a leafcutter garden for long. This is where the biology becomes, I think, quietly astonishing in a way that is hard to overstate without overstating it. The ants manage contamination through a system that operates at multiple levels simultaneously, behavioral, chemical, and microbial. And the three levels interact in ways that researchers have been working to characterize for several decades, with the picture still gaining resolution as better analytical tools become available. At the behavioral level, ants continuously weed the garden. Workers move through the fungal mass removing foreign growth, carrying contaminated material to waste chambers, adjusting airflow through the garden structure by physically rearranging the substrate. The weeding is constant, it never finishes. The garden is a living system in continuous flux, and the maintenance behavior matches that flux in real time. Below the behavior, chemistry. The same metapleural secretion that protects individual ant bodies is applied to the garden. Workers groom the fungal surface with the same mouthparts they use to groom nestmates, distributing antimicrobial compounds across the cultivated mass. The garden, in a real sense, receives the same chemical attention as the ants themselves. It is part of the colony's body in the way that matters biologically. It is something the colony's chemistry actively maintains. And then below that, something that was not discovered until relatively recently, and that shifts the picture considerably. The ants carry bacteria. Specifically, they carry bacteria from the genus Pseudonocardia, actinomycete bacteria that live on the surface of the ants' bodies in specialized crypts on the cuticle that appear to be anatomical structures evolved specifically to harbor these organisms. The bacteria produce antifungal compounds, antibiotics, in the functional sense of the word, compounds that suppress the growth of the particular mold, Escovopsis, that most threatens leafcutter gardens. The ants have a bacterial partner living on their bodies, maintained across generations, passed from worker to worker through contact, that has co-evolved with the colony to produce chemistry targeted at the colony's primary agricultural pathogen. Just take a moment with that. In the past, before this was known, the metapleural gland was understood as the primary source of antimicrobial chemistry in leafcutter colonies. That understanding was not wrong, and the gland remains important. But it was incomplete. The ants were running two systems simultaneously, one internal and one external, one glandular and one microbial, and the external system was living on their bodies in structures that had been anatomically visible without anyone knowing what they were for. The discovery changed the framing, because now the question is not just how ants produce antimicrobial compounds, It is how ants maintain a living pharmaceutical partnership across colony generations, passing beneficial bacteria from ant to ant through social contact, keeping the bacterial population viable and active and producing the compounds the colony needs without any understanding of what the bacteria are or what the compounds do. It just works. The bacterial community on a leafcutter ant's cuticle is not static. It varies across the colony's population, differs between castes, shifts in composition in response to the infection pressure the colony is experiencing. There are researchers who have found evidence that the bacterial community adjusts, that when Escovopsis pressure increases, the ants that carry the most effective bacterial strains are preferentially groomed and their bacteria more widely distributed through the colony's social network. The colony is, in some functional sense, managing its microbial partners as actively as it manages the fungal garden. Both are agricultural acts, in the broadest meaning of the word. Both involve maintaining a living system for the colony's benefit. Both involve behavioural and chemical investment at a scale that, when you look at it directly, sits somewhere between complex and extraordinary. I tend to find the bacterial partner story more surprising than the fungal garden story, though I'm not entirely sure why. Perhaps because the garden is visible. You can see it, photograph it, hold the concept in mind without difficulty. The bacterial community is invisible, distributed across thousands of bodies, moving through the colony as a living chemical resource that the ants carry without knowing they carry it. It is small and everywhere at once. Now the pacing changes, because the chapter has reached the part where the clusters of detail need to thin, where the biology has been laid out and what remains is to sit in it quietly. The garden is somewhere below ground right now. It smells, according to researchers who have described it, of something organic and faintly sweet, the metabolic output of a living fungal mass being continuously tended in a warm, humid chamber. Workers move through it without pause. The garden grows in the particular way it has grown for 50 million years, its structure shaped by the ant's behaviour and the ant's behaviour shaped by its needs. On the bodies of those workers, bacteria are producing compounds. On the garden surface, those compounds are being applied. In the substrate of the garden, Escovopsis spores that arrived on a returning forager are encountering a chemical environment they cannot easily survive. Not because anyone planned it. Because the colony that runs this system more effectively than the alternatives is the colony that is here. The garden continues. The bacteria continue. The ants move through both, carrying chemistry they don't know they carry, maintaining a system whose full architecture they will never need to understand in order to operate it with extraordinary precision across time spans that make the word ancient feel insufficient. There is another layer to this story. Below the garden, in the waste chambers where contaminated material is carried and kept, the biology is doing something that is not quite disposal and not quite containment. It is something that took researchers a long time to look at carefully. That is where the next chapter goes. There is a pressure that never stops, not physical pressure, chemical pressure. The continuous, directionless, impersonal pressure that pathogens exert on every host population they encounter, not through intent, not through strategy, but through the simple fact that the ones that survive contact with the host defences are the ones that reproduce, and what they pass on is whatever allowed them to survive. Generation after generation, pathogen populations shift toward whatever works, whatever gets through, whatever finds the gap in the chemistry that the host has been building across its own generations. This is the arms race that every living system with a pathogen is running, not as a choice, as a consequence of existing. The ants have been running it for longer than almost any other insect lineage with a comparable social structure, which means the pathogens that target them have also been running it for that long, which means the gap between offence and defence in this particular relationship is not a gap that closes, it moves, It shifts. One side gains ground and the other adjusts. And what you see in the present moment, the metapleural secretion, the bacterial partners, the behavioral repertoire of grooming and isolation and removal is not a finished system. It is a current position in something ongoing. Think of it like a geological formation. What you see when you look at a cliff face is not the rock's final form. It is where erosion has reached so far. The process that shaped it is still running, too slowly to observe directly, producing changes that will only be visible across spans of time, much longer than any observer's presence. The cliff looks stable. It is not stable. It is just changing more slowly than perception can track. The ant colony's chemical defenses look stable in the same way, but the metaphor needs to be set down now because it has done what it can do, and the biology deserves to be met on its own terms. The specific pathogen that has been most studied in the context of ant chemical resistance is Metahysium, a fungal genus that infects a wide range of insects and that has been co-evolving with ant lineages long enough that different species of Metahysium show different degrees of virulence across different ant species, a pattern consistent with the kind of host-specific adaptation you would expect from a long-running co-evolutionary relationship. The ants that have been in contact with Metahysium longest tend to show the most robust chemical defenses against it. The defenses that work are the defenses that were tested. This is not a reassuring fact exactly. It means the robustness of the current system is partly a measure of how much the system has already been challenged. The ants that have the most effective antifungal chemistry are the descendants of colonies that encountered the most effective fungi. The chemistry is calibrated to a threat that calibrated itself against the chemistry. It's not entirely clear how stable that calibration is at any given moment. What is clear is that resistance in ant colonies is not purely chemical. The behavioral components of the defense system shift in response to infection pressure in ways that suggest the colony has something like a dynamic response capacity, not fixed behaviors, but behaviors whose frequency and intensity adjust to the current threat level. When Metahysium exposure increases in experimental settings, grooming rates increase. Isolation behaviors appear sooner and more reliably. The threshold for self-removal by infected individuals appears to lower. The colony tightens its response without any individual ant having assessed the situation. This dynamic quality is part of what makes the system durable across evolutionary time. A fixed chemical defense is a fixed target. A behavioral system that adjusts its intensity is harder to fully circumvent, because circumventing it requires the pathogen to overcome not just a specific compound but a responsive system that increases its investment when challenged. The cost of that responsiveness is real, though. Heightened grooming takes time and energy. Isolation removes functional workers from the colony's labour pool. The bacterial partners that produce antifungal compounds on the cuticle are metabolically maintained. The ants invest resources in keeping those communities viable, resources that are not available for other uses. Every component of the defense system runs on energy that the colony has to acquire, allocate and spend. There are moments in colonies under sustained high infection pressure where the cost becomes visible. Growth slows, foraging range contracts, the colony pulls inward, investing more in defense and less in expansion, making the kind of trade-off that any system with limited resources makes when one demand increases suddenly. The colonies that survive this are the ones that set the trade-off point correctly for the specific pressure they are under. Set it too conservatively and the infection spreads. Set it too aggressively, and the colony starves its other functions into failure. The biology finds the point that works and holds it there, approximately, imprecisely, in the way that all biological regulation is approximate and imprecise and nonetheless functional. It's worth sitting with that imprecision for a moment, because it runs counter to the way biological systems are often described, as elegant, as optimized, as refined to a precision that human engineering can only admire. The reality is messier than that. The ant colony's defense system is not optimal. It is good enough under the conditions it evolved in most of the time. It fails in edge cases. It is outpaced by novel pathogens it has no evolutionary history with. It costs more than it would need to if the pathogen were not also adapting. That is what the arms race produces. Not winners, not optimization. A continuous, costly, imperfect equilibrium that looks like stability from far enough away. Your own immune system works in something like the same way. Not elegant, not optimal, but durable across the conditions it evolved in, expensive to run, and vulnerable to threats that fall outside its evolutionary experience. The parallels between mammalian immune function and ant colonies' defense have been noted in the literature carefully, with appropriate caveats about the differences in mechanism. The parallels are real, so are the differences. Both systems are running an arms race they cannot win and cannot stop running. The ant colony below ground is in the middle of that race right now. The pathogens it faces are the current generation of organisms that have been shaped by contact with ant defences across millions of years. The ant colony's defences are the current generation of behaviours and chemistries shaped by contact with those pathogens across the same span. Neither side knows the other exists. Both sides are entirely defined by the other's presence. The pressure never stops. The colony keeps running. And in the part of the nest that hasn't been visited yet in this narration, in the waste chambers, in the places where the colony puts what it cannot use and cannot allow to stay, the pressure is managed in a way that is less about chemistry and more about something stranger and quieter, something the next chapter moves toward without hurry. There is a chamber at the periphery of the nest, not the centre, not where the queen is, not where the larvae are kept, not where food is stored or processed or distributed. At the edge. Down a tunnel that diverges from the main traffic corridors in a direction that most of the colony's workers never travel. The waste chamber. Every ant colony produces waste. Dead nestmates. Exhausted fungal substrate. Rejected food material. The shed exoskeletons of molting larvae. Fungal contaminants removed from the garden, and all of it, with a consistency that holds across species, across geographies, across nest architectures that vary enormously in their other features, gets carried to a specific location and kept there, not scattered, not abandoned wherever it falls. Carried, deliberately, in the functional sense that ant behaviour can be called deliberate, to a place that is not where anything else happens. You start to notice when you look at waste management in ant colonies for long enough that the behaviour is more structured than disposal needs to be. The geometry of it is specific. The location of waste chambers relative to brood chambers, to food stores, to the nest entrance, these relationships are not random. They follow patterns that across species suggest the placement is doing something beyond simple removal. The research on this took a while to catch up to the observation. What the placement is doing turns out to involve airflow, temperature gradients, and microbial ecology in ways that make the waste chamber less of a dump and more of a managed zone. Ant nests are not sealed systems. Air moves through them, along gradients created by the temperature difference between the warm interior and the cooler soil at the nest's margins. Waste chambers are consistently positioned in locations where this airflow pattern carries volatile compounds, the gases produced by decomposition, the spores released by contaminated material, away from the brood and food stores rather than through them. The ants did not design this. The colonies that placed waste in locations where airflow carried pathogens toward the brood produced fewer surviving offspring. The colonies whose waste placement, by whatever combination of behavioural tendency and nest architecture, happened to route contamination away from vulnerable chambers, those colonies persisted. The behavior and the architecture co-evolved around a physical fact of fluid dynamics that the ants have never needed to understand. Still, there is something worth pausing on here, in the gap between what the behavior looks like and what it is. From inside the nest, if observation were possible at that scale, the workers carrying waste to the periphery would look purposeful, directed, as though they know where they are going and why. And in a narrow behavioral sense, they do know where they are going. The chemical gradient that guides them to the waste chamber is a real signal, a real navigation cue, and they are following it accurately. What they don't know is why that location was selected by the colony's history to carry that signal. The reason is millions of years old. Inside the waste chamber, the biology continues without any ant attending to it. The decomposition of organic material proceeds through microbial action, bacteria and fungi breaking down the physical and chemical complexity of dead tissue, shed cuticle, rejected substrate into simpler compounds. This process produces heat. It produces moisture. It produces volatile gases and, in the case of contaminated material, potentially viable spores of the very pathogens the colony is trying to manage. The chamber is, in other words, a concentration point for exactly what the colony cannot afford to spread, and the ants manage it, not by sterilizing it, not by chemically neutralizing it in the way a disinfectant would, by positioning it where the nest's own physical dynamics contain the risk, and by applying even to this peripheral space the same behavioral maintenance that the rest of the nest receives. Workers do enter the waste chamber periodically, and they do apply grooming behavior and metapleural secretion to the walls and the accumulated material, though at lower frequency than the brood chambers receive. The differential is meaningful. The brood chamber receives near constant chemical attention because its occupants are the colony's most vulnerable members and its most essential investment. The waste chamber receives periodic attention because complete neglect would allow contamination to build to levels that the airflow management alone could not contain. The colony calibrates its maintenance investment to the risk level of each space, not through any central assessment of risk, but through the aggregate behavior of workers responding to local chemical gradients. More chemical signal of vulnerability draws more maintenance behavior. Less signal draws less. The waste chamber generates a different chemical profile than the brood chamber, and the colony's response reflects that difference automatically. What stands out most in the waste chamber biology, when you read through the studies on it, is how much the space is doing passively, how much of the contamination management is handled not by ant behavior, but by the physical and microbial properties of the space itself, the temperature, the airflow, the existing microbial community in the waste substrate, which includes organisms that compete with and suppress the pathogens the colony is most concerned about. The waste chamber has its own ecology, not cultivated in the way the fungal garden is cultivated, not tended, not maintained with the same behavioral investment, but shaped over time by the consistent deposition of particular materials in particular conditions into a microbial community that does something useful for the colony without the colony directing it to do so. This happens in your gut, in aversion. Microbial communities shaped by the consistent environment your digestive system provides, doing things that are useful for your body without your immune system directing them, suppressing pathogens through competitive occupation of the same physical space, producing compounds that your own cells use, existing in a relationship with your biology that took evolutionary time to establish, and that disrupts in specific documentable ways when the environment that shaped it changes. The parallel is not exact, but the principle that a managed environment develops a biology of its own that then contributes to the management appears in both places, at very different scales, in very different organisms across very different spans of time. The waste chamber is quiet right now. A worker enters from the main tunnel, carrying something small and pale, a fragment of spent fungal substrate from the garden above. It moves to the far wall, deposits the material with the precise brief movement that characterizes ant transport behavior, and turns back toward the entrance without pause. The chamber receives what it receives. The airflow continues in the direction it has always continued. The microbial community in the substrate adjusts, incrementally, to the addition of new material, the way any living community adjusts to a change in resource availability, not dramatically, not immediately, but continuously in the direction that the chemistry allows. Below this chamber, or beside it, depending on the nest's architecture, there is another space. One that the colony uses differently, not for what has been discarded, but for what the colony cannot yet classify, cannot yet determine how to handle. The biology there is less settled. It is where some of the colony's most interesting edge case behavior occurs, in the narrow zone between what the defense system can manage comfortably and what begins to exceed it. The next chapter moves into that zone. There is a point in the operation of any defense system, biological, chemical, behavioral, where the thing doing the protecting begins to affect the thing being protected, and the line between the two stops being clear. This is not a failure exactly. It is a structural feature of systems that operate through broad-spectrum action rather than surgical precision, systems that work by creating conditions hostile to a wide range of threats, and must therefore create conditions that are, to some degree, hostile to everything in that environment, including the organisms the system is meant to serve. The metapleural secretion is a broad-spectrum antimicrobial. That word, broad-spectrum, carries a particular weight in the context of this chapter, because it means the compounds the gland produces do not distinguish between harmful fungi and beneficial ones, between the pathogen that would kill an ant and the microbial community on the garden surface that the colony has been cultivating and depending on for 50 million years. The chemistry acts on fungal cell walls and membranes through mechanisms that are, at the molecular level, indifferent to which fungus they are encountering. Think of it like a thermostat set slightly too high. The room stays warm, reliably warm, warm enough that the cold never quite gets in, but warm enough also that certain things that prefer cooler conditions begin to struggle, and the struggle is not visible until it has been happening for long enough to show up in the output of whatever those things were producing. The leafcutter fungal garden is sensitive to the metapleural secretion in exactly this way. Researchers working with leafcutter colonies noticed, in experimental conditions, that when metapleural secretion was present at elevated concentrations in the garden environment, concentrations that arose when colony stress increased grooming rates and therefore increased chemical deposition, the cultivated fungus showed signs of suppression, not dramatic collapse, subtle thinning, a slowing of growth in localized areas where the chemical concentration was highest. The kind of change that would be easy to miss if you weren't looking for it, and that the colony would need to manage without being able to identify its source. The ants are doing this to their own garden. Not always, not typically. The normal operating concentration of metapleural secretion in the nest environment is calibrated by evolutionary selection to a level that suppresses pathogens effectively while remaining below the threshold that significantly damages the cultivated fungus. The calibration is not perfect. It holds within a range. And when the colony comes under elevated pathogen pressure, when infection events increase grooming rates and the chemical deposition rises, the garden begins to experience concentrations that push against the upper edge of what it can tolerate. The system is protecting itself from one thing while inadvertently stressing another. Your immune system does this in a way you may have experienced directly, though you probably didn't think of it in these terms at the time. Inflammatory response. The cascade of signaling molecules and immune cell activity that your body generates in response to infection or injury is a broad-spectrum process. It creates conditions hostile to pathogens, but those conditions are also hostile to your own tissues. Prolonged or excessive inflammation damages the cells it is nominally defending. The joints that swell and ache during certain infections are experiencing immune activity directed at something else, collateral effects of a system that cannot quite confine its action to the intended target. The principle is older than vertebrates. It appears in different molecular forms across the full range of organisms that have immune systems of any kind, because the problem it represents, how to act broadly enough to be effective without acting so broadly that you damage what you're protecting, does not have a solution that doesn't involve trade-offs. Now the pace of this chapter slows, because the most interesting part of what happens next is not dramatic. The leafcutter colony, under elevated pathogen pressure, increases its chemical defenses and stresses its garden, and then, in a behavioral adjustment that researchers have documented but not yet fully explained at the mechanistic level, something shifts. The ants modify their grooming behavior in the garden specifically, not in the brood chambers, not at the nest entrance, but in the garden, where the sensitive cultivated fungus is. The intensity of grooming in the garden decreases relative to grooming elsewhere in the nest, even as overall grooming rates remain elevated. The colony is, in some functional sense, protecting the garden from its own defense system. How this adjustment is regulated, what chemical signal or behavioral cascade produces the differential, is one of the open questions in this area of research, and the openness of it is genuinely interesting rather than frustrating, because it points at a level of behavioral sophistication in the colony's response that was not anticipated when the basic metapleural system was first characterized. The colony is running a regulation system on top of a defense system, not through any individual ant's assessment of the situation, through the aggregate behavior of a population responding to multiple simultaneous chemical gradients, the gradient that increases grooming in response to pathogen signal, and something else, some additional signal from the garden that pulls grooming behavior back below the threshold that would damage it, the two signals producing in their interaction an output that neither would produce alone. What that second signal is, precisely, is still being worked out. It may be a compound produced by the cultivated fungus itself when it is under chemical stress, a distress signal of sorts, not directed at the ants, not produced for that purpose, but detectable by the ant's sensory system and capable of modifying behavior in a way that happens to benefit the fungus. Coevolution, producing a communication channel that neither party designed. Or it may be something else. The research is ongoing and the picture is incomplete, and that is, right now, the honest state of things. The garden continues to grow, slower in some areas than others, thinner where the chemical concentration has been highest, recovering in the places where the grooming intensity has pulled back. The ants move through it with the same continuous attention they've always given it, their behavior adjusted in ways they cannot be aware of, responding to signals they will never need to understand. The defense runs, the regulation runs alongside it. The garden exists in the overlap between them, in the narrow band where the chemistry is hostile enough to protect and restrained enough to allow growth to continue. Outside that band, things go wrong. Not often. The band is wider than it might seem from this close. The colony has been operating within it for long enough that the biology has found the tolerances and learned, in the only way biology learns anything, to stay within them, most of the time. The edge cases, the moments when the band narrows, when pathogen pressure spikes and the chemical response overshoots and the garden thins in ways that take real time to recover, are where the colony's resilience is most visibly tested and where the next chapter finds its question, not about failure, about how close to the edge the normal operation of this system actually runs, and what that proximity means for a colony that cannot afford to find out where the edge is by crossing it. Touch is the first sense, before light, before sound, before any of the more elaborate sensory systems that evolution eventually produced in various lineages. There was contact, one surface meeting another, a physical exchange of information that required no interpretation, no processing delay, no distance between the signal and the receiver. The ant lives almost entirely in that world. Its antennae make contact with surfaces dozens of times per second during normal movement, with nest walls, with the ground, with nestmates passing in the opposite direction along a tunnel corridor, with the bodies of larvae it tends, with the chemical residue left by every other ant that has moved through the same space in the hours before. Each contact is brief. Each contact transfers something. You already know this feeling at a level that doesn't require explanation. The way a handshake tells you something before any word is spoken. The way a familiar surface, a doorknob in your own home, a particular texture you've touched 10,000 times, registers as known before you've consciously identified it. Touch carries information that bypasses the longer processing routes, arriving somewhere in your nervous system as a kind of immediate knowing. The ant's antennae are doing that, continuously across every surface they encounter. But what they are reading is more specific than texture, more specific than temperature or moisture, though those qualities are part of what the antennae detect. What they are reading primarily is chemistry, the hydrocarbon profile of whatever surface is being touched, a chemical signature as specific to each ant as anything biological can be, encoding information about species, colony membership, caste, age, reproductive status, and recent history of exposure. The cuticle carries all of this. It carries it not in a structure analogous to memory, not in a place where information is stored and retrieved, but in its chemistry itself. The waxy outer layer of the cuticle is a record of what the ant has been in contact with, what compounds have been transferred to its surface through grooming, what environmental exposures have left residue on its body. It is a kind of living document, continuously updated, that every nestmate reads through antennal contact. When an ant has been exposed to a pathogen, that exposure changes the document, not through conscious alteration, through the chemistry of the exposure itself, the compounds produced by the pathogen, the changes in the ant's own surface secretions in response to infection pressure, the subtle shifts in the hydrocarbon profile that accumulate as the body begins to respond to something it is working to contain. These changes are small, they are real, and they are readable by the antenna of nestmates before any visible symptom appears. This is the biological basis for what earlier chapters described as the colony's early detection capacity. It is not mysterious. It is contact chemistry, operating through mechanisms that are well characterized at the molecular level. The mystery, if that word belongs anywhere in this, is not the mechanism but the scale at which it operates. A colony of hundreds of thousands of individuals, each one reading and updating the chemical document of every nestmate it touches, producing a distributed record of the colony's health state that no single ant holds and every ant contributes to. The information is in the network, not stored anywhere specifically, present everywhere the network is active. You carry something like this in your body's immune memory, not in a single location, but distributed across populations of cells that were shaped by past exposure and that carry the record of that exposure in their receptor profiles, their surface chemistry, their readiness to respond to particular molecular signatures. The record is not centralized. It is embodied in the literal sense in the biology of millions of cells whose individual characteristics encode the history of what the body has encountered. The ant colony's chemical record works differently at the mechanistic level, but the distribution is similar. The absence of a central storage location is similar. The fact that the record is continuously updated through ongoing biological activity, rather than fixed at the moment of encoding, is similar. Both systems carry history in their present state. The cuticle of an old forager, one that has been outside many times, that has encountered many surfaces, that has been groomed by many nestmates and has groomed many in return, carries a chemical complexity that reflects all of that contact, not as memory, as residue, as the accumulated layering of every transfer that has occurred across that stitched surface over the ant's working life. A younger ant carries less. The nurse ant that has spent its entire life inside the nest carries a chemical profile shaped almost entirely by the nest interior, by the brood chambers, by the queen's pheromones, by the specific chemical atmosphere of the space it has never left. Its cuticle reads differently from the forager's. Both are readable. Both tell a story to the antennae that encounter them, a story told in hydrocarbons rather than words, perceived in milliseconds of contact rather than across the duration of a conversation. The colony reads these stories constantly, without pause, without rest. In the dark, in the tunnels, in every brief contact between bodies moving in opposite directions along the same narrow corridor. There is something settling about this, once you let it sit. The colony does not need a brain to hold its collective knowledge. It does not need a central record. The knowledge exists in the ongoing activity of the system itself, in the contacts, in the transfers, the continuously updated chemistry of every individual surface. Remove the activity and the knowledge dissolves. Sustain the activity and the knowledge is always current, always distributed, always available to any ant whose antennae make contact with any other. The system is the record. An ant that has been isolated from the colony for long enough begins to lose the chemical markers of colony membership. The hydrocarbons that identify it as belonging to a particular nest to gradually shift. In the absence of constant renewal through nestmate contact, away from the colony profile. If it returns after extended isolation, nestmates may respond to it with aggression. The recognition signal has degraded, and the colony's chemistry reads the returning ant as something unfamiliar. The identity was never internal to the ant. It was maintained by the network. This is where the chapter settles and stays, not with a conclusion. With the image of an ant moving through a tunnel, antennae in continuous contact with the wall, reading the chemical residue of every ant that has passed this way before it, adding its own residue as it moves, contributing to the record that the next ant will read, in an exchange of information so continuous and so quiet that it has no beginning you could point to and no end that is not the end of the colony itself. The cuticle carries what the colony needs it to carry. Below the cuticle, inside the body, the chemistry of the ant is doing something that this chapter has only touched the edge of. Something about how the infection signal moves from the surface into the interior, and what the interior does with it when it arrives. That is where the next chapter begins to look. Somewhere in the 14th century, the city of Ragusa, now called Dubrovnik on the Adriatic coast, began requiring ships arriving from plague-affected regions to anchor offshore for 30 days before passengers could disembark. 30 days, then 40. The period extended as the practice developed, the word quarantine arriving eventually from the Italian for 40, a number chosen not through epidemiological calculation, but through a combination of biblical precedent and accumulated observation that isolation reduced transmission in ways that, at the time, no one could explain mechanistically. It worked, imperfectly, partially better than nothing. And it was, by any measure of human medical history, a significant development. The recognition that separating the potentially infected from the definitely healthy produced outcomes different from not separating them. That physical distance between bodies could interrupt whatever invisible process was turning sick people into sources of more sick people. This was the 14th century. The ants had been doing it for somewhere between 30 and 60 million years. Not the same behavior, not with the same mechanism, not in response to the same pathogens, but the functional principle, remove the potentially infected individual from the population of healthy individuals before transmission can occur, was operating in ant colonies across geological epochs, while the continents were still moving toward their current positions, while the mammals that would eventually produce humans were still small and largely nocturnal, while the word disease had not yet been thought in any language by any mind that existed. I find it difficult to treat this as a simple fact. It keeps requiring a moment. Not because it makes the ants intelligent in the way we use that word, not because there is anything intentional in what they do, but because the functional convergence between a behavior that emerged from human observation and reasoning across centuries of epidemic experience, and a behavior that emerged from evolutionary pressure across tens of millions of years of colony-level selection, produces a kind of double image that is hard to look at directly and easy to let blur into something less precise than it actually is. The precision matters. The ant quarantine, if that word is allowed here used carefully, operates through mechanisms entirely different from any human isolation practice. It is chemical. An ant whose infection load has reached a detectable threshold emits a surface signal, a shift in the hydrocarbon profile of its cuticle that nestmates read through antennal contact and that triggers behavioral responses in both the infected individual and the ants around it. The infected ant may begin to withdraw, not through decision, through the alteration of its own behavioral outputs by the compounds the infection produces. Some pathogens, particularly certain fungal species, produce chemicals that modify the behavior of the host in ways that serve the pathogen's reproductive interests. But in the early stages of infection, before the pathogen has fully established, the behavioral change may reflect the ant's own immune response rather than pathogen manipulation. The internal chemistry shifts. The behavioral outputs shift with it. The ant moves toward the colony's margins. Nestmates may assist this movement. They groom the infected ant with an intensity that is, in the early stages, an attempt at decontamination, applying metapleural secretion to the cuticle surface, attempting to interrupt the infection before it progresses beyond the surface. But as infection advances and the chemical signal strengthens, grooming behavior transitions. The physical contact continues, but the character of it changes, becoming less about application of protective compounds and more about movement, ants nudging, redirecting, physically guiding the infected individual away from the brood chambers, away from the food stores toward the periphery. The transition between these two behaviors is not abrupt. It is a gradient, driven by the intensity of the chemical signal producing a response that escalates as the situation escalates, without any ant having assessed the situation in any way that resembles assessment. Research on this behavioral sequence has been conducted across multiple species using experimental infection protocols that allow researchers to observe the colony's response at controlled stages of pathogen progression. What the data shows is a consistency across species that suggests deep evolutionary origin. This is not a behavior that evolved recently in response to recent pathogens. It is old. It is conserved. It appears in ant lineages that diverged from each other tens of millions of years ago and have been evolving independently since. The behaviour was present before most of the species it now operates in existed. That is the time-layered quality of this chapter's biology. The behaviour predates the ants that currently express it in the same way that your body's apoptosis pathways predate you, conserved from ancestors so distant that the evolutionary relationship requires genetics to confirm, running in your cells through molecular machinery laid down before anything you could recognise as your lineage had appeared. Old solutions to old problems. The problems changed. The solutions were modified around their conserved core. What remains is functional, tested, deeply embedded in the biology of organisms that have been shaped by the specific pressure of living in dense social groups where infection can spread through contact. Dense social groups with constant contact between individuals are, from an epidemiological standpoint, extremely high-risk environments. The ant colony has been managing that risk since before the risk was a concept anyone had formed. The city of Ragusa eventually stopped requiring ships to anchor offshore as the mechanisms of disease transmission became better understood and more targeted interventions became available. The practice that worked when the mechanism was unknown became less necessary once the mechanism could be addressed more directly. The ants have not developed more targeted interventions. They don't need to. The behavioural system that emerged under selection pressure tens of millions of years ago remains sufficient for the pathogens it evolved to manage. It fails against novel pathogens the colony has no evolutionary history with. Introduce species laboratory exposures to fungi from geographically distant regions, the way any evolved defense fails against threats it was not shaped by. But within the ecological context the colony actually inhabits, the old system holds. It holds right now. Somewhere in the colony, an ant is moving toward the periphery. Nestmates have been grooming it with increasing frequency. The chemical signal on its surface has been shifting for hours, imperceptibly at first, then more distinctly, and the colony's response has been escalating in proportion. The brood chambers are deeper in the nest. The distance between them and the infected ant is increasing. That distance is the whole point. That distance is what 30 days of anchored ships offshore was also trying to produce through an entirely different mechanism, arrived at through an entirely different process, separated from the ant's behaviour by tens of millions of years, and the full cognitive distance between a city council making a public health decision and a colony of insects running a chemical response to a surface signal, both producing the same outcome: the infected individual separated from the vulnerable population by whatever means the biology or the culture had available. The outcome is the same. The mechanisms could not be more different. And somewhere in that gap between mechanism and outcome, in the space where two entirely unrelated biological and cultural processes arrive at the same functional solution, there is something that I tend to sit with for longer than the science requires. Not a conclusion, just a noticing. The colony is still moving. The ant near the periphery is still. The distance holds. Inside the body of that ant, below the cuticle where the chemical signal's been broadcasting the colony's concern, the infection is doing what infections do, progressing through the internal biology of a small body with limited internal defenses, consuming resources, producing compounds, following the logic of a pathogen that has been refined by the same evolutionary process that refined the defenses trying to contain it. What happens inside that body, in the cellular and chemical space where the ant's own biology meets the pathogens, is where the next chapter looks, quietly, without urgency, at what the internal environment of an infected ant actually contains, and what it is still, even at that stage, attempting to do. There is a number that appears in the population biology of social insects that tends to get absorbed too quickly when encountered in passing. a number that deserves more residence time than it usually receives because it points at something about the nature of collective biological systems that is easy to miss if you move past it toward the more dramatic facts on either side of it. The number is not a threshold for catastrophe, it is a threshold for function. In a mature ant colony, the proportion of workers that can be lost to predation, to infection, to the ordinary attrition of foraging in a world full of hazards, before the colony's overall function begins to measurably degrade, is smaller than most people would estimate if asked to guess. The colony looks robust from outside. It looks like a system with so many components that losing some fraction of them would barely register. The reality is more specific than that, more sensitive than that, and the sensitivity is not a weakness in the system, so much as an accurate reflection of how tightly the colony's chemistry and behavior are calibrated to its current population composition. The margin between a functioning colony and a struggling one is narrower than the colony's apparent size suggests. This chapter is interested in what sits inside that margin. Not the catastrophic end of the range, where infection overwhelms the colony's defences and the population collapses in a cascade that researchers can induce experimentally and have documented in the field after exposure to particularly virulent novel pathogens. That end of the range is real, and it matters, but it is not where most colonies spend most of their time, and it is not where the most interesting biology lives. The most interesting biology lives in the narrow operational band just inside the margin, where the colony is managing elevated pathogen pressure, where the defense systems are running at higher intensity than baseline, where the trade-offs described in earlier chapters are being actively made, and where the outcome is not yet determined. Think of it like barometric pressure before a weather change. The atmosphere is doing something, the instruments show it clearly, but whether the storm arrives or passes depends on factors still in play, still interacting, still capable of going either way. Then set that image down, because the biology doesn't need it. The colony under elevated infection pressure is running multiple systems simultaneously, and the interaction between them is where the margin is managed or lost. Grooming rates are higher, which means more metapleural secretion is being distributed, which means the fungal garden may be experiencing suppression at its margins, which means the colony's food production is potentially affected, which means the energy available for continued elevated grooming is potentially reduced. The loop is real. It is circular. It is the kind of feedback dynamic that makes biological systems difficult to model cleanly, because each variable is also a function of several other variables, and the relationships between them are nonlinear in ways that don't resolve into simple predictions. The colony navigates this without modelling it. Through behaviour that responds to local chemical gradients, through the aggregate output of hundreds of thousands of individuals each doing something simple, the colony finds a path through the feedback loop that keeps the system functional, not optimally, not without cost, but functional. The grooming intensity adjusts, the foraging allocation shifts, the garden receives modified attention. The chemical deposition in sensitive areas is regulated by mechanisms that are still being characterized in the research literature. The colony threads the margin, most of the time. There are colonies that don't. That encounter a pathogen load too heavy for the current defense capacity, or a chemical imbalance that the regulatory mechanisms can't correct in time, or a sequence of environmental stressors that reduces the colony's population below the threshold where the defense system can maintain its coverage. These colonies decline. The process is not sudden. It is the gradual failure of one calibration after another. The grooming coverage becomes insufficient. The chemical signal becomes too weak at the nest entrance. The bacterial partners on the cuticle thin out as the worker population thins. The garden begins to show more contamination than the reduced workforce can remove. The margin narrows. Then it closes. This happens. It is part of the biology. It is not the exception that proves the rule, it is the mechanism by which the rule is enforced. The selection pressure that keeps the surviving colonies calibrated to the threshold that works. Your own body has margins of this kind, and you've probably felt them, though not always recognized them as such. The weeks when you are sleeping poorly and eating inconsistently and under sustained stress, when the immune system's surveillance is maintained, but its response capacity is reduced, when you catch things you would normally fight off without symptoms, when recovery takes longer than it should. You are in the margin. The system is functional. The calibration is slightly off. Most of the time, the demands on the system don't exceed its reduced capacity and you don't notice. Sometimes they do, and the illness that follows is the margin closing. The body recovers and the margin reopens usually, because recovery mechanisms exist that are themselves calibrated to restore the system to its normal operating range. The ant colony has equivalents: population recovery through the queen's continued egg production, behavioral flexibility that allows the colony to shift resources toward defense during elevated threat periods and away from it when the threat subsides, the resilience of the bacterial partner communities on the cuticle that can recover from chemical suppression when the suppression eases. The margin is not fixed. It breathes in a sense, expanding and contracting with the colony's population, its nutritional state, the seasonal variation in pathogen pressure, the local ecology of the specific soil environment the nest inhabits. A colony that is marginal in late summer, when foraging demands are highest and the nest population has been depleted by months of attrition, may be well within its functional range by the following spring, when new workers have been produced and pathogen pressure has shifted with the season. Time is part of the margin. This is worth staying with, because it changes the framing of what a colony under stress actually is. Not a system approaching failure necessarily. A system in a particular phase of its annual cycle, running closer to its limits than it will in other phases. Managing the demands of that proximity through behavioral and chemical adjustments that are part of its normal repertoire. The stress is real. The proximity to the margin is real. And the colony's capacity to manage that proximity is also real, encoded in the same biology that encodes everything else, in the glands, in the behavior, in the social network of chemical communication that keeps the system coordinated across hundreds of thousands of individuals who will never meet each other in any way that resembles meeting, but whose aggregate behavior produces something that functions from the outside like a coherent response to a coherent assessment of a situation. The assessment is not coherent. It is distributed and chemical and automatic. The response somehow is coherent anyway. Right now, in a colony somewhere under pressure, it has been managing for weeks. The margin is holding. The grooming continues at its elevated rate. The bacterial communities on the cuticle are producing what they produce. The garden is thinned at its margins and recovering in the areas where the chemical intensity has eased. The waste chamber receives what it receives. The workers near the periphery attend to the individuals whose chemical signal requires attention. The queen, deeper in the nest than any of this, continues to produce eggs. The eggs will become workers. The workers will join the population that manages the margin. The margin will have more to work with. This is the colony's answer to proximity, not to move away from the edge, but to keep producing the components that maintain its distance from it, continuously without pause in the dark. The edge is still there. It is always there. What keeps the colony from it is not distance. It is the continuous operation of every system described across the previous 13 chapters: the chemical atmosphere, the grooming network, the glandular secretion, the bacterial partners, the behavioral cascade that removes what cannot be managed, the waste placement that contains what cannot be removed, the regulation that prevents the defense from consuming what it is defending. All of it running simultaneously in a colony that does not know it is doing any of it. The margin holds. For most colonies, on most days, in most seasons, the margin holds. And in the final chapter, the narration moves away from the edge entirely, not to a place of resolution because biology doesn't resolve, but to a longer view, a quieter place, where all of this has been running for so long that the word ancient stops feeling adequate and something else is needed. Something that doesn't quite exist yet in the language available for it. That is the year Alexander Fleming noticed something unusual on a culture plate in a laboratory in London. A mould had contaminated a bacterial culture, and around the mould the bacteria were absent. The zone of clearance was small. The observation was not immediately understood. But Fleming recognized that the mould was producing something, secreting something into the surrounding medium that was preventing bacterial growth in its vicinity. He called the active substance penicillin. What followed from that moment is one of the most consequential developments in the history of medicine. Antibiotics changed what it meant to survive an infection. They changed surgery, changed childbirth, changed the trajectory of diseases that had been killing people reliably for centuries. The discovery was real. The impact was real. The science that grew from Fleming's observation and the work of the researchers who followed him deserves every part of the recognition it has received. And underneath all of it, beneath the laboratories and the culture plates and the clinical trials and the decades of pharmaceutical development, something had already been working. Not penicillin specifically, not the same compounds, not the same molecular targets, not the same organisms, but the same principle that a living system could produce compounds that suppress the growth of harmful microorganisms in its environment, had been operating in ant colonies for a span of time that makes 1928 feel like it happened this morning. It is strange to notice this. Strange in a quiet way, not a dramatic one. Not because the ants knew something humans didn't, not because there is any competition between these two facts, but because it raises a question that doesn't have a clean answer, the kind of question that sits at the edge of sleep rather than the center of it. How much is already working right now in systems we haven't looked at carefully yet? The history of biology is substantially a history of noticing things that were already happening. Cells were already dividing before anyone had a microscope. DNA was already encoding proteins before anyone had sequenced a genome. The immune system was already running its surveillance before immunology existed as a discipline. The discovery is always of something that predates the discovery by a span of time that the discovery itself then makes visible. The ant colony's antibiotic system is part of that history, but it is also something slightly different from most of the examples in that history, because it was not discovered by studying ants directly at first. It was discovered obliquely through the observation that ant colonies maintained remarkably low infection rates despite living in soil environments that are extraordinarily rich in fungal and bacterial diversity, through the question that observation prompted, through the slow accumulation of evidence that pointed toward the glands, the secretions, the bacterial partners, the behavioral system that distributes the chemistry. The discovery followed the anomaly. The anomaly was always there, a colony in a field in soil that contains thousands of fungal species, in tunnels that are warm and humid and organically rich, exactly the conditions that should, by any reasonable expectation, produce catastrophic infection rates, maintaining itself with a cleanliness that the environment around it does not predict. Someone noticed that it didn't fit, and the noticing led eventually to everything this video has been describing across 14 chapters. Now, the colony is still in the field, or a colony like it, in soil like that, the same chemistry running in the same glands, The same bacteria on the same cuticle surfaces, the same behavioral cascade distributing the same compounds through the same social network of grooming contacts and antennal touches in the dark. Nothing about the colony changed when the science found it. The system was not waiting to be understood in order to function. It was not incomplete before the researchers arrived. It was running at the same fidelity it has always run, calibrated to the same pressures it has always been calibrated to, producing the same outcomes it has been producing since before there was anything on this planet that would eventually become curious about it. That is the part that takes a moment, not the complexity of the system, not the elegance, the indifference of it to being observed. Living systems do not perform for observers. They run because the ones that ran are the ones that left descendants, and the ones that left descendants are the ancestors of everything currently alive, and everything currently alive is therefore the accumulated output of an unbroken chain of functional biology stretching back to origins so distant that the planet itself was different then. The ant colony is one current expression of that chain. So are you. Your cells are running chemistry tonight that predates every observation ever made of it. Your immune system is doing things that were characterized in the 20th century and things that are still being characterized now, and probably things that haven't been noticed yet by anyone looking. Your mitochondria are running a process that was already ancient when the first animal cell formed. Your body is full of biology that was working long before anyone thought to ask what it was doing. This is not a comforting thought exactly, but it is a settling one, somehow. The sense that the working is not dependent on the knowing, that the biology continues in its own competence regardless of whether any mind has yet found it and named it and traced its mechanism, that you are being maintained right now by processes that do not require your understanding of them to maintain you. The ant colony doesn't know what it knows. Neither do you, fully. Neither does the science yet. There is more biology in the world than has been observed. More chemistry running in more organisms in more ecological contexts than any current research program has reached. The soil under a single square meter of forest floor contains more microbial diversity than has been fully characterized anywhere. The chemical interactions between species in any given ecological community are more numerous than the studies conducted on them. The glands of insects, the bacterial communities on arthropod cuticles, the volatile compounds exchanged between organisms in ways that modify each other's behavior. These are areas where the research is active and the picture is incomplete, and the incompleteness is not a problem to be solved so much as a condition to be inhabited. The biology will keep running while the picture is completed. It ran before the picture was started. The colony is underground right now. The air in the tunnels carries the chemistry it has always carried. The metapleural glands are secreting. The grooming contacts are occurring at the frequency the colony's current state requires. The bacterial partners on the cuticle are producing what they produce. The fungal garden, if this is a leafcutter colony, is being tended by workers whose behavior is calibrated by signals too faint to measure with most current instruments. The queen is producing eggs. The eggs will become workers. The workers will groom and forage and tend and carry waste to the periphery and return through the entrance to be greeted by antennae that read their chemical history in the fraction of a second of contact. And the colony will continue, as it continued before Fleming's culture plate, as it continued before the first human noticed that infected people sometimes infected other people and began tentatively to act on that observation. As it continued before any mind on this planet had formed the category of infection, or the category of defense, or the category of medicine. The chemistry was already working. It is still working. In the dark, without any need for this to have been said. If you made it all the way here, thank you. Genuinely. These videos take a long time to put together. And knowing that someone listened through to the end, or fell asleep somewhere in the middle, and let this run quietly while they rested, means more than I can say simply. If the sleepy biologist is something you want to keep coming back to, subscribing is the simplest way to make sure the next video finds you. And if you felt like tonight's journey was worth something, a like, a comment about where you were listening from, or a super. Thanks if you're feeling generous. Those things go directly into making the next one possible. The comments are always open. People leave the most thoughtful things there, and I read everyone. Sleep well. Your body is doing remarkable things tonight without asking anything from you at all. See you next time.